Next up, we have Marie Olland, a Ph.D. student in environmental engineering. She'll be presenting her work on forensic chemistry, detecting chemicals and biosolids. Marie, the stage is yours.
Thank you, Eric, and thank you everyone for being here today. As Eric said, today I'll be talking about biosolids, specifically identifying the chemicals in biosolids and taking the toxic ones out. So remember this diagram? As we heard, biosolids are the solid waste that comes out of the wastewater treatment plant. So they get treated and they leave. And biosolids have some pretty cool benefits when mixed into soil. They help lock up carbon, which can help reduce our carbon footprint. When mixed into agricultural soil, they're super nutrient rich and so can help increase our crop yields. And if we actually use them, then they're not going to landfills, which is where about half of our biosolids go in some places. Sounds pretty cool, right? Unfortunately, biosolids also have a couple downsides, one of which is they are filled with chemicals. So think along the lines of personal care products like soaps, deodorant, toothpaste, pharmaceutical products like antibiotics, medication, anything that stormwater runoff picks up, like heavy metals and pesticides. Basically, anything that goes down a drain, any drain, has chemicals in them, and they end up in our wastewater treatment plant. And if they don't get treated properly, they end up in our biosolids. And so you can imagine all of these chemicals in our biosolids is a problem, especially if we're going to be growing food out of this soil. And I'm not trying to scare you, because they're not all necessarily harmful, and some of these chemicals do get treated and removed. But which chemicals and to what degree, we don't know. The good news is that's what I'm working on. My first research question is better understanding what chemicals are actually in these biosolids. And how do I do this? I use the same instrument we heard in Lorinda's talk, a liquid chromatograph and mass spectrometer. And this allows me to separate and detect the tens of thousands of chemicals in biosolids. The method I use is called non-target analysis, which lets me detect everything in these samples. And what's great about this method is it doesn't limit my search, but unfortunately the way this instrument works is it fragments my chemicals into pieces. And so I don't have any chemical names associated with these fragments, and I have to do a little detective work. Basically, I get this huge file with masses, retention times, and intensities for each of those individual fragments. This file, for example, had 80,000 chemical fragments. And this gives me a much more complete picture of what's actually in these biosolids. But as I said, we don't know what chemicals these fragments belong to. So what I can do next is compare these masses and retention times to databases to get a list of chemicals that were probably in my sample, or what we call suspected chemicals. Chemicals that fraction the same way or have the same retention time are probably the same chemical. And so here, for example, we can see the black fragments that we measured from the instrument line up very well with the red chemical from the database. And so we can be pretty sure this chemical was in our samples. Here, not so much. And so now I have a list of all the chemical fragments in biosolids and a slightly smaller list of suspected chemicals in biosolids. Which brings me to my next research question. How do we actually remove all of these chemicals? And is our removal method effective? The approach I'm using is bioremediation, which is just the use of a biological agent, in my case bacteria, to remove these pollutants in a more natural and cost-effective way. The way I do this is by loading a syringe with bacterial cells and this gelatiny substance. I slowly release this mixture, a few drops at a time, into a vat of cross-linking solution. This solidifies the beads with the cells inside of them, giving them extra protection. Then I make lots of soil pots to actually test these beads. I have a pot of soil alone and biosolids alone as baselines, a pot of mixed soil and biosolids, and a pot of soil, biosolids, and those beads. And then I let them do their thing. Over time, the bacteria literally eat away at the chemicals. I'll sample every couple weeks, and after a couple months, they'll have done their thing, and I can analyze what has happened. I'll do the same chemical analysis, get the same big sheet, and run statistical tests on those resulting suspect and fragments lists, on their patterns and trends in their chemical composition. This work is still in progress, but here's an example of what that might look like. Samples that are very similar or have the same chemicals in them should cluster together. So ideally, we'll see the biosolid samples cluster together, and then the soil samples making their own separate cluster because of their different chemical compositions. And then hypothetically, we should also see the pot of the soil and mixed biosolids fall somewhere in between those two. And then if treatment was successful, we should see a completely separate cluster for the samples that came with the pots with the beads, indicating some sort of chemical change in that pot. And then I can use the suspected chemicals list to actually figure out which of those chemicals went away with treatment, if any increased or didn't go away over time that we should pay extra attention to. Some other cool things I can do with this data is look at DNA from those same samples. I can get a better idea then of which bacterial strains are removing which specific chemicals. I can also run toxicity tests on some of those chemicals that either increased over time or didn't go away that are of particular concern. But in the end, all this helps us determine how much risk we're actually introducing by mixing our soil and biosolids, and potential ways of naturally reducing these chemical risks, so that hopefully we can safely and sustainably reuse our biosolids without putting chemicals in our food and environment. Thank you.
Thank you.